Okay, good morning. So the first thing we're going to do is go over at least parts of this worksheet. The real point of this is not just to see how editing works, but to get practice looking at the genetic code and predicting what the changes will be with different editing events. So you need to have a copy of the genetic code handy to be able to understand these things. Okay, so the first question on the worksheet is that one form of A to I editing occurs preferentially at the central A of codons of the type CAN, where N can be any one of the four. Now, okay, this is a slightly artificial situation, but we're just going to go with the constraints. So we have a CAA, CAG, CAC, and CAT as possible editing sites. So what we have to do is look at the genetic code and indicate what the initial amino acid was before the editing event, and then look to see what it's been changed to as a result of the editing event. Okay, so first of all, we've got a CAA, look at the genetic code. Now we have to get used to the single letter code where it's not GLU, it's Q stands for glutamine. So when we edit it, we have a CIA where I is inosine. It's read as CGA by the translation machinery. So now we look at the genetic code and see what CGA encodes, and we can see that it's arginine, which is R. Second one, same idea, CAG, also reads as glutamine. CIG reads as CGG, which is also arginine. CAC reads as histidine, looking at the genetic code, which is H. And CIC, after the editing event, reads CGC. Again, that's arginine. Okay, last one is CAT, is a histidine codon if you look at the chart. CIT reads as CGT, which is also arginine. So that's a fairly straightforward example of how this editing thing works. Okay, the second question is even simpler, actually. We've got a small part of the DNA sequence encoding a sodium ion channel, along with the genome-encoded protein sequence. Now, this does point out something important in terms of recognizing these things. That remember that the cDNA is created by copying the final mRNA. So when you compare the cDNA sequence, what you're really looking at is an edited sequence if it's edited. And so comparing that to the genome sequence will tell you what the original was before the editing one. Okay, for this, that's sort of an aside, but comparing cDNA to genomic is a way to see these things. Okay, so we've got this methionine here, which is an AUG or ATG codon, and we're told that it's replaced by a valine. Well, so we take our ATG, there's only one A in it, change that to ITG, which is the inosine, and that's read as GTG. So we can look at the genetic code for GTG, and we can see that it encodes a valine. Here's our genetic code. Okay, so the HIV rev sequences, these are actually fairly simple because they're basically giving you the answer. They're giving you a series of amino acids that are encoded by the genome, and they're told what the editing event produces. So again, this is, more practice at reading the genetic code than it is anything else. So the first example, we have a TAT, and we're told that the A has been edited, and that we look and see what that means. We see that TAT is a tyrosine codon, just as they're telling us. And then we edit that, which gives you TAT goes to TIT is read as TGT, and the genetic code says that that is in fact histidine. So in this, what they're telling us is what the answer should be, and it's just practice to looking at the genetic code to convince yourself that that's actually true. Okay, so the next bits are a little bit harder. You have to get used to the single letter code again and the nomenclature, but you're not told what the original codon was. So you actually have to write out the various possibilities in order to figure out what's happened. And be aware some of these have unique answers, but others have more than one possibility. Okay, so I'm not going to do all of these, but I will do a few important ones. Okay, so in terms of nomenclature, this S367G, what that means is that at position 367 in the protein, the original codon reads serine. And that after the editing event has occurred, that codon will now read as a glycine. So the way to approach this is we have to look at the serine codons, because we don't know which serine codon it is. And so we write out all the possibilities. Now there's 6 serine codons. Fortunately for us, the last 3 don't have any A's in them. So we know that that's not the event, because we're always going to be editing A's for this. So that leaves us with 3 serine codons, which are the possible first codon. So now we have to look at these and see how they could be edited such to produce a glycine. So we do them one at a time. AGT can be changed to IGT, read as GGT. Okay, that is in fact a glycine codon if you look at the genetic code. Similarly, AGC can be changed to IGC, read as GGC. This is also a glycine codon, so that one could work also. TCA can be changed to TCI, read as TCG. This is not a glycine codon. So either of the first 2 possibilities could be what the editing event was. We don't know what the answer actually is, but it's certainly one of those 2. Okay, here's another one. Isoleucine 342 methionine is isoleucine and methionine change at codon 342. So we look and see the isoleucine codons. Those are ATT, ATC, and ATA. Methionine is only one codon, that's ATG. So now we modify the isoleucine codons in all the ways that are possible with this type of editing. So we start with our ATT, that gives us a GTT, that's a possibility. ATC gives us GTC. Well, neither one of those is methionine codons, right, because we're looking for an ATG. Our ATA, however, gives us an ATI, gives us an ATG, right, which is actually what we're looking for. So we would say that the ATA created the A modified, well, the last nucleotide modifier would give us an ATG, and so that's certainly what would have happened. Okay, if we modify the first nucleotide, that gives GTA, which is not a methionine codon. If you modify both sites, you get GTG, which is also not a methionine codon. So the only one that works is that the original was an ATA, which has now been modified to give you ATG. Genetic code again. Okay, so I think everybody can continue and do the rest of those pretty much exactly the way I did them. Okay, so we're going to continue talking about RNA editing, a little more detail about the A to I type of editing, which is the adenosine deaminases, and then just a few words about another type, which is Cdu editing, and that's carried out by apobec enzymes. Okay, so reminder, here's adenosine. If it's converted to inosine by removal of the amino group, you now have a double bond O at that position, and that inosine will base pair differently from what the adenosine did. So here we have a adenosine base pairing with uridine in its usual way. Inosine, however, base pairs with cytidine, which is to say it acts as if it were a G. Okay, a few other things about these substrates and what they look like. The Adar enzymes recognize double-stranded RNA substrates, that they don't recognize a single strand, and these pictures show them base pairing. So in the examples we've seen, what's being edited is a base paired structure, which includes an exon where the actual amino acid modification is going to be, and an adjacent intron. So the exon actually base pairs in an imperfect way with the adjacent intron. So in this case, we've got exon 11, and here we have exon 5. And with the result, that modification does change the amino acid sequence. Okay, so they use double-stranded RNAs. All right, so this will occur in the nucleus before splicing, because obviously the intron is still present. Here's a better picture of one of these, where there's a base paired structure involving exon 5 and intron 5. But notice it's an imperfect base paired structure. So it's still not quite possible to predict what's going to be edited or what, but they're going to be these sort of partially mispaired double-stranded RNA structures of one sort or another. Okay, so I now need to point out that there are actually two types of Adar editing. The ones we've been talking about are the site-specific variety, where you have specific protein sequences that are changed as a result of the inosines. So those are site-specific in that it's a very particular A in a very particular mRNA to change a very particular amino acid. The other type is called promiscuous editing, which is actually far more prevalent and involves, again, long double-stranded substrates, but they occur not in protein coding regions, but either in introns or in 3' untranslated regions of RNAs. We'll see more about that on a later day. Okay, a little other things about the Adars. It is a gene family. In human, we actually have three. We've got Adar1, Adar2, and Adar3. The two Adar1s that are shown here are protein isoforms that are created by different splicing of the mRNA. So they're the same gene, but they produce two different proteins, similar. In Drosophila, there's only one Adar, which is primarily modifies targets in the central nervous system. Squid, which do a lot of these, squids and octopuses do a lot of editing. They have the Adar1, also the two copies, or two different versions of Adar2. So it's a very widespread gene family, and it seems to be common among most metazoans, if not all of them. Okay, so what distinguishes the members of these family primarily is, remember the idea of protein domains, where different parts of the protein can actually serve different functions. So with ADARs, we have, of course, adenosine deaminase activity, and that activity resides in the what's called the catalytic domain of the protein. So that's the business part of it, the actual catalytic region. Okay, but the other ones, they also have to bind RNAs. So ones that act on RNAs have double-stranded RNA binding domains, which are shown in green in the picture. So other adenosine deaminases, such as the ones that act on tRNAs, don't have these double-stranded RNA binding domains. Human ADAR1 also has a different region, additional region, called the Z binding region, and these bind to nucleic acids in the Z conformation. So what the Z conformation is, these are regions of the nucleic acid where it's helical, but the helical is left-handed and not right-handed. Okay, so the ones that act on tRNAs have only the deaminase domains, no double-stranded or no Z binding domains. Okay, I think I probably just said this. Okay. ADAR1 is actually primarily important in the immune system, but we'll get to that on a different day. Okay, ADARs and human disease. Okay, ADARs and lack thereof have been associated with a lot of human diseases, including many neurological disorders. It's considered very particularly important in the brain, in nervous tissue, but also in cancers and immunological disorders. The cancer situation is kind of hard to understand because sometimes there's a problem, or a change anyway, in editing in the positive direction and sometimes in the negative direction. And it's hard to know what's cause and effect with these things sometimes. So hypoediting is a case where there's not as much editing as you would expect, but in other cases you get too much editing, hyperediting. So this is obviously an area of importance that people are trying very hard to figure out. Okay, so the second type of editing we're going to only talk a little bit about, which is Cdu editing. It's the same idea. We have cytosine deaminases, and what they do is they remove the amino group from the cytosine, which creates uracil. And of course, uracil base pairs differently from cytosine. The AU base pair will base pair like an AU base pair, not the original GC base pair. So enzymes that catalyze these reactions are called ApoBec, and there's a whole family of them for us. Okay, here's what cytosine deaminase is. Here's the cytosine base. It's got an amino group. It's deaminated to produce a double bond O at that position, which is uridine or uracil. So the deaminase changes a C to a U, which will of course now base pair like a U with an A, not with a G. Okay, so the first known example of this in proteins at least is the Apolipa Protein B, and how it behaves differently, how the mRNA behaves differently in liver versus intestine. Okay, so this is Cdu editing. Okay, what looks like in the DNA, there's a glutamine codon, a particular glutamine codon, which is edited, or can be edited, to produce a stop codon, a UAA stop codon. So this is an in-frame glutamine codon, which is going to be altered in the RNA to form a UAA stop codon. So what this looks like, this is at codon 2153 in the Apolipa Protein mRNA. Okay, in the liver, the RNA is not edited, and the mRNA is translated to form a nice long protein, 4,563 amino acids. That's a big one. And it functions in transport of cholesterol. And that's in liver. In the intestine, however, the RNA is edited at one particular position, where there's a glutamine codon at 2153. It's edited to form a UAA stop codon, with the result that the Apolipa Protein B that's produced in the intestine has only 2,152 amino acids. It's a functional protein. What it does is absorb lipids from the intestine, but it's obviously a very different protein from the one that's produced in the liver. Okay, so that's a CDU change at codon 2153, and that does change the function of the protein. Okay, so in mammals, there are lots of other CDU editing events, but those are not, the protein coding region ones are extremely rare. Mostly they're in things like 3' untranslated regions, but very few actually influence protein sequences. In plant organelles, however, it's a different situation. The CDU editing is actually very prevalent, and there are lots of events that actually do change the protein sequences. Okay, so there are several hundred editing sites in mRNAs in mitochondria and chloroplasts, among the RNAs that are encoded by the organelle. So these editing events can change or destroy start and stop codons, can change the amino acid sequence, they can occur within introns, and they can also create silent mutations. So it's very prevalent in plant organelle genomes, but very relatively less frequent, certainly, in human. Okay, so the real importance of apobecs in human is very likely targets of DNA. This used to be kind of considered sort of obscure, but it turns out that as a result of editing, these apobec enzymes have essentially antiviral properties, and they cause mutations, and they're part of the first line of defense, part of our host innate immune system. They're also important in cancer. I'm just going to show you very briefly one short example. Okay, so recognize that this is actually a large family of proteins that have different targets. So it's going to be involved in host defense against viruses in the immune system. Okay, so here's kind of a brief overview of the HIV life cycle and what apobecs can do to it. Okay, so first we're going to have an infection of a sensitive cell by this virus. Okay, so what's going to happen is the virus binds to the surface of the sensitive cell, releases its capsid inside the cell, is uncoated, and begins the reverse transcription process, which is the first step in integrating into the chromosome to become part of the host cell DNA. Okay, the apobec enzyme is already there in the cell, some of it introduced in the virion itself, and what it will do is create mutations as in the process of the reverse transcription. It also directly impedes reverse transcription as part of its life cycle. Okay, the other thing so it's going to create mutation. The other thing it does is manage to get itself, to some extent, packaged into new virions. So after the reverse transcription occurs, the provirus is there in the nucleus, and it's going to be creating mRNAs to create new virions so that the infection can proceed. And it's going to package those mRNAs into new capsids by biting off the surface of the cell. Well, apobec is sneaky and manages to get itself into the virion some of the time so that it's ready to create mutations when it gets into the new cell. So the apobec enzyme, first of all, interferes with the reverse transcription process, and it also creates mutations that can inactivate the new virus. Okay, so remember, it's going to be a double-stranded DNA, which integrates into the host cell genome. produces viral RNA using the host cell RNA polymerase, and then will be used to create new virions. And that will, of course, lead to the packaging of virion of genome-length RNA into new virions, which will then take some of the apobec protein with it, so that when it goes into the new cell, the apobec is ready to actually create mutations in the virus as it tries to replicate. So here's one picture of this. We've got the apobec going into the new virion as it buzzes off the cell surface. It infects a new cell. And just representative, we've got some G codons here, which will be reverse transcribed to form CC. But then these can be mutated by the apobec or deaminated by it to give a U. So the ultimate effect of that is to take essentially a GC codon and convert it to an AU or AT codon. So that results in mutations in the virus. So that way it can start, right? Right. The mechanism is actually opposed by an HIV protein. So the virus is able to fight back. And people are trying to find inhibitors of VIV so that the apobec can work better. But don't worry about that part of it. So this is just an experiment to demonstrate that the apobec actually can mutagenize this virus. So what they had is just cells that were overexpressing the apobec enzyme, and then parts of the HIV genome sitting there as targets. So all the little dots indicate where mutations did occur as a result of this apobec overexpression. So it is definitely a mutagenic enzyme relative to this virus. Okay, so don't worry about this either, but what it's showing us, the results of an experiment in which you had cell lines that were overexpressing apobec to show that this could have a deleterious effect on any viruses trying to infect the cells. So starting with a vector-only part, we have, normalizing to 1, that if you have cells that are overexpressing the apobec enzymes, it greatly reduces the infectivity of the viruses that are being produced. MLV is a different virus which is less sensitive. So this is a definite, strong antiviral enzyme, and people are very interested in how these work and getting them to work for us better.